Hello everyone, welcome to another lessons learned video. Today we're going to discuss the ventricular system and the flow of cerebrospinal fluid. So within the brain we have these blue spaces, which you can imagine as empty spaces. They're not really empty, they have fluid inside of them. And what lines the inner walls of these spaces, the ventricular spaces, is a tissue called the choroid plexus, which is going to secrete this cerebrospinal fluid that flows through them. So the CSF, or the cerebrospinal fluid, will flow this way. So it will flow like this over here. Now this general big structure over here, this is the lateral ventricle, and there's going to be two of them. There's one on the right side, and then there's one on the left side. So we have two lateral ventricles. And the CSF flows basically from the posterior horn, from the lateral horn, to the body and the anterior region. So this is, should have labeled it. It's going to then flow here through this space, which is called the interventricular foramen of Monroe. Then it is going to enter this area, which is called the third ventricle. And there's going to be, excuse me, there's going to be one third ventricle, so it's unpaired. And it's going to be formed by the thalamus. So the walls of the thalamus and the diencephalon in general are going to form this third ventricle. Then the CSF will flow through this area, which is referred to as the cerebral aqueduct. Sometimes you might see it referred to as mesencephalic aqueduct because it runs through the midbrain, which midbrain also is called mesencephalon. And then this space over here is called fourth ventricle. It's kind of in between the cerebellum and the pons. So fourth ventricle. Then after fourth ventricle, it will run through here and enter the central canal of the spinal cord. It can also run through the apertures, which are these holes, and enter the subarachnoid space around the brain. If it goes down to the central canal, it can come out through the cauda equina, which is the tail of the spinal cord, and go to the subarachnoid space around the spinal cord. So let's explain what happens after. So if I take out this lateral ventricle, so you have space, and also this. If you were to, I haven't drawn all the layers obviously, but if you were to look at some of the meningeal layers here, this is what you'd see. You'd see skull, right? So this white layer is skull. Underneath that you'd have the endoosteal layer of the dura. In the sagittal area of the brain, basically on the midline on top, you will have kind of this protrusion into the longitudinal fissure of dura mater. That's going to be called falx cerebri. And inside the falx cerebri there's the superior sagittal sinus. So the sinuses are basically just very large veins that carry deoxygenated blood away from the brain. So they carry it into the internal jugular veins, and this is right here the superior sagittal sinus. Then underneath that is going to be the let's do don't know what to pick. Let's just do like green. It's going to be the meningeal layer of the dura. But it's not actually green, obviously. But underneath that we have the arachnoid. And then underneath the arachnoid is the subarachnoid space, which has cerebrospinal fluid flowing through it. So that's where the CSF will go when it exits the apertures. And then we have the pia underneath that. The pia we can see red because it's highly vascularized and it's going to be very closely attached to the entire surface of the cortex. Enlarge this a little bit to see it better. When the CSF flows through the subarachnoid space around the brain, it's going to reach the area where it's going to meet, like it's going to be over here. And when it's around the area where the superior sagittal sinus is, we're going to have these channels called the arachnoid villi. So the arachnoid villi, I'm going to erase part of this to represent the arachnoid villi. It looks kind of like this. It's a channel between the superior sagittal sinus and the subarachnoid space. And it allows cerebrospinal fluid to flow from the subarachnoid space into into the superior sagittal sinus, and then that's how it exits the whole area in the nervous system, right? It exits and then goes to circulation. Now the arachnoid villi, what they do not allow to happen is for deoxygenated blood to enter into subarachnoid space, right? This does not occur. So they're one-way channels. That's important because if you were to allow this, then the concept of a blood-brain barrier would not make any sense. So the tissue the tissue that's going to secrete cerebrospinal fluid is the choroid plexus, which is on the inner walls of all of the ventricles. So all of these ventricles have choroid plexus within them. And the cells in there that make, monitor, and secrete CSF are called the ependymal cells. They keep the composition and the rate of production of CSF fairly constant. It's independent of most other things like blood pressure and intracranial pressure. So if there's a deviation in the composition of CSF, so for example, if someone gets a lumbar puncture and their CSF has too much proteins, that might signal that there's a viral infection or a viral meningitis. Could also have a bacterial meningitis if there's not a lot of glucose in the CSF, because then the bacteria is just eating the glucose. Because the choroid plexus cells, the ependymal cells, are just going to secrete CSF at the same rate all the time, any obstruction in the flow of CSF is not going to cause them to slow down their rate of production. So that can cause issues, because then the pressure, there's more fluid in the ventricular spaces than is allowed, so you would have too much pressure, on the walls of the ventricular spaces, which would then push to the brain, and then basically all the interstitial spaces in the brain would compress to try to allow for that fluid to be there. And that condition is called hydrocephalus. So any obstruction of flow within the ventricular spaces that would not allow enough CSF to leave through these arachnoid villi would cause hydrocephalus. So I hope you learned something there. And thank you for watching.